Hello, everyone. My name is Malia, and the topic of my podcast assignment is titled Powering Purple Prokaryote Production by Jesse Noor. To begin with, the purpose of the purple phototrophic bacteria produce certain kinds of wastewater along with electric current to produce valuable products such as hydrogen without much waste. Jesse begins his podcast with discussing a bacteria named Streptomyces, which is a chain-like fungus. Streptomyces was discovered in 1958 by Edinger. Streptomyces can produce various antibiotic compounds like carbamazepine. Jesse Noor begins to then discuss wastewater and its association with purple bacteria. Wastewater is any water that has been affected by human use. Usually we treat wastewater as a waste. We try to remove other contaminants from it and other undesirable stuff to make it safe for their environment or make it usable as water again. Depending on what type of wastewater it is and what's in there, can be a lot of chemical energy contained in it, which could be gathered for energy or used to power some other purpose in microbes, which are great in gathering every little bit of energy from their environment. There are some around in the world that can get any little energy there is from different organic compounds, or even from minerals, or from light from photosynthesis. According to Daniel Poole, now researchers have found a way to reduce carbon emissions from sewage and produce hydrogen energy at the same time. Their method uses purple bacteria and electrical currents to capitalize on the organic materials we flush down the toilet every day. There have been some researchers who have tested how purple phototrophic bacteria can be used to produce hydrogen and other valuable components from wastewater. The anoxygenic phototrophs grow under anaerobic conditions in the presence of light and do not use water as electron donor as higher plants. They grow autotrophically with CO2 and hydrogen or reduced sulfur compounds, act as an electron donor. Dr. Bryant says that the purple bacteria were the first bacteria discovered to photosynthesize without having an oxygen byproduct. Instead, their byproduct is sulfur, which has been demonstrated by first establishing the bacteria's reaction to different concentrations of oxygen. It was found that bacteria moved quickly away from even the slightest trace of oxygen. Then a dish of bacteria was taken and a light focused on one part of the dish, leaving the rest dark. As the bacteria cannot survive without light, all the bacteria moved into the circle of light, becoming very crowded. If the bacteria's byproduct was oxygen, the distances between individuals would become larger and larger as more oxygen was produced. But because of the bacteria's behavior in the focused light, it was concluded that the bacteria's photosynthetic byproduct could not be oxygen. Jesse then starts to discuss what it is that purple bacteria can produce, which is hydrogen gas, and how that gas can be used to generate electricity. The purple prototrophic bacteria use an energy from infrared light, and they have a complex metabolic system and are highly electroactive. The purple prototrophic bacteria are not photosynthetic in the same way as plants that produce oxygen in their reaction, but they can absorb light energy to use to power themselves, and they can use this in combination with organic compounds if they're available. They have a versatile metabolism that can be tailored in multiple purposes. If you give them a nitrogen fertilizer, they can make a protein-rich feed for animals. If you limit the nutrients that they can get, they can make a lot of biopolymer. And a biopolymer is an organic molecule that are composed of repeating monomers and are produced by living organisms. The biopolymer can be used as a biodegradable plastic. If you give the purple prototrophic bacteria carbon without nitrogen, it can make a hydrogen gas, which can be used to generate electricity too. It is possible to feed the purple bacteria electrons directly from an electrode with electric current, which is called electrosynthesis, using electrons instead of photons or chemical energy. In this particular study, the researchers took wastewater and put it in an environment with infrared light for photosynthesis and volatile acids to feed the microbes that are growing or trying to grow. So this was best for growing purple phototrophic bacteria that could produce hydrogens. They also observed the success of these microbes by looking at the specific wavelengths of light and that they can absorb because they are particular colors, which is why they're called purple. In particular, the compound called bacteria chlorophyll A makes them look the way they do. Chlorophyll A absorbs the wavelengths of violet, blue, and orange light and functions as a primary electron donor during the electron transport chain in photosynthesis. They detected how many bacteria there are by how much is present and by how much of the particular wavelength of light that they absorb. The researchers used this enriched culture of bacteria for their experiments. They are looking specifically just at the hydrogen production. And Jesse then goes on into including nitrogen into the hydrogen gas production and other acids that better the hydrogen production. This hydrogen is produced in the process of nitrogen fixation. Nitrogen fixation is the chemical process by which atmospheric nitration is assimilated into organic compounds, especially by certain microorganisms, as a part of the nitrogen cycle. So in the process of nitrogen fixation, the enzymes that are involved in the N2 gas add protons and electrons to it to make two whole molecules of ammonia, which is a colorless gas with the characteristic pungent smell. It dissolves in water, giving a strongly alkaline solution. Ammonia is also NH3. So that is 3 protons for every nitrogen, and this can be used for protein production, which includes nitrogen and also nucleic acids. And as a byproduct of this process, they produce hydrogen, which has 2 protons stuck together, and so they make at least 1 molecule of this gas for every molecule of nitrogen gas. If you give the cells of ammonia or other nitrogen compounds that are already produced, it could prevent this process by shutting off the enzyme that does it. They tested that by giving them nitrogen compounds, they actually found that a low concentration of ammonia enhanced the production of hydrogen, whereas a higher concentration made it stop and go and get rid of it. They found that in a controlled atmosphere of nitrogen gas, the cells didn't produce that much hydrogen they could detect. This is because most of the electrons were going towards fixing the nitrogen instead of producing hydrogen. The hydrogen that did produce was probably being reabsorbed by the cells to use energy so that it is not wasted. But if the cells were in an atmospheric argon gas, which doesn't react with the enzyme, which produces the hydrogens, the cells keep trying to fix the nitrogen. But there's no nitrogen around, so the electrons and the protons they would use for it just make hydrogen. Argon and a low amount of ammonium turned out to be the best for hydrogen production. They also tried an amino acid called glutamate instead of ammonium. Glutamate is a salt or ester of glutamate acid. They observed that the glutamate enhanced the growth of cells. It also didn't inhibit the hydrogen production, which is really good. So even in high amounts, it doesn't turn off the nitrogen fixing enzyme. It just helps the cells grow better. It is also helpful by minimizing the production of carbon dioxide, and instead the cells took in carbon dioxide and made it into more cells. So they decided to use a large concentration of glutamate for their further experiments. They tried enhancing the production by adding electrons directly, which turned out to work even better. They tried different carbon sources like malic acid, butyric acid, and acetic acid. The malic acid turned out to be the best out of all three, which allowed the growth in hydrogen production, which allowed most growth in hydrogen production. They found that the advantage of the electrode and bioelectric process was that the cells captured all the CO2 that was made. Jesse then discusses that what is used in the making of electricity, which is microbial fuel cells, and how the electrons produce hydrogens, which makes this very useful in the making of electricity for this process by using microbial fuel cells, where the microbes themselves generate electric current of just use of solar panels. It should be possible to modify the conditions and the flow of electrons to change the outputs to different desired configurations to make hydrogens. So it is a versatile system. In some cases, this can actually be very useful. It does rely on specific conditions for obtaining value from less valuable starting materials. And that if its conditions is not met, then it is more difficult to put in more value. And Jesse then concludes his podcast by letting his audience know that the entire process is best, most easier when making energy and producing maintainable waste in the result of having a better cycling of resources. So therefore, there has to be a balance of value of the product with the cost of inputs and the cost of processing. But there's a lot of potential work. There's a lot of potential work so that it works well, or at least better than anything else that has been used for extracting energy from this process. And it is a good step towards developing more sustainable streams of production and waste so that we can have a cycle of resources instead of a directional flow from putting them out of the ground and putting them back into the ground as a waste, because we don't want to use up all of our resources. So in conclusion, basically to summarize this entire video is just introducing what it is that purple phytotrophic bacteria does and what it produces, and which is the certain kinds of wastewater along with electric current to produce valuable products such as hydrogen without much waste.